Alright, in this video I wanted to follow up on our discussion of mitosis, and in this video we'll discuss meiosis, a similar cell division process, but that gives us quite a different result. So let's go ahead and talk about meiosis. So sometimes this will get referred to as the meiotic phase, like we talked about the mitotic phase for mitosis, but most often I'll simply refer to this as meiosis. When we talked about mitosis, we made a simple declarative statement about what the products of mitosis are. We want to make that same statement for meiosis, and that way we can compare the outcome of mitosis versus meiosis. So when I talk about meiosis, I say this. I say meiosis produces 4 genetically different gamete or haploid cells. All right, so meiosis produces 4 genetically different gamete cells. So how is that statement different than what we declared for mitosis? So you might remember that in mitosis, we produce just 2 cells, not 4. So that's one difference. Meiosis produces 4 cells, mitosis produces 2. You might recall that when we talked about mitosis, we said that every cell we make is genetically identical to the other cells. Here we're making genetically different cells. So that's another difference between the two processes. And then you might remember that during mitosis, we said that we make somatic or diploid cells. Here we're making gamete or haploid cells. So that's the third difference between these two processes. 4 cells versus 2 for mitosis. Genetically different cells versus identical cells for mitosis, and then gametes, which are haploid cells, versus somatic cells, which are diploid for mitosis. So we're going to go through the steps of meiosis, and at the end we'll think about why these 4 differences occur in meiosis when comparing to mitosis. Alright, so before we talk about the specific steps of meiosis, let's set a little groundwork. So the first is that meiosis, just like mitosis, follows interphase. So remember, interphase involves the steps G1, the G1 phase, it involves S phase, and it involves G2 phase. So these 3 steps, G1, S, and G2, they are also occurring prior to meiosis, just like they did for mitosis. So after G2, instead of going into mitosis, we're going to enter these meiosis steps. The names are going to look familiar, things like prophase, metaphase, anaphase, telophase. But then we're going to double the steps. So we're going to get prophase II, metaphase II, anaphase II, and telophase II. So we're doubling up the steps, and at the end we're going to have these 4 gamete cells that will stop at that point. They're not going to divide ever again. So we're going to use those cells. Unlike in mitosis, where once we've produced our 2 cells, they both enter back into G1 phase. So meiosis is not a cycle like mitosis is. All right? Okay, so one other reminder before we talk about these specific steps of mitosis, and that is that DNA replication occurs prior to meiosis. Remember that DNA replication takes place during interphase, when we go from this single chromatid structure of a chromosome to these chromosomes that have 2 sister chromatids attached to one another, and they have this more X-shaped structure. So before DNA replication, we're 1 chromatid. After DNA replication, we're 2 sister chromatids attached together. Remember that happens during S phase of interphase. So S phase, remember that S stands for synthesis of DNA. So during interphase, we do DNA replication. During S phase, remember S phase is synthesis of DNA. And that's why we get these chromosome structures that are 2 sister chromatids attached to one another. So now, remembering those details, we can discuss the steps of meiosis that result in producing these 4 genetically different gamete cells. All right, so what are the stages of meiosis? Again, all of these names are going to sound very familiar after learning about mitosis. We're going to go through 8 steps that I've shown here. We're going to go through Prophase I, then we'll go through Metaphase I, then Anaphase I, then Telophase I. After Telophase I, we repeat these steps over again, and in the 2 cells that we have after Telophase I, we're going to go through Prophase II in both of those cells. Then they go through Metaphase II. Then the next step is Anaphase II. And we follow up with Telophase number 2. The only other point I want to make here is to remember that telophase is always associated with a cytokinesis event. So following Telophase I and Telophase II, we have cytokinesis. And that's how we go from 1 cell to 2 cells after Telophase I, and from 2 cells to 4 cells after Telophase II. All right? So we're going to go through each of these stages. We're going to point out the major things that are happening, and we'll think about the results at the end. So we did this for mitosis as well. I'll point out the major points. Along the way, I have some bolded and underlined points that I want you to pay special attention to. Anything I have bolded or underlined, I think is a critical difference that meiosis has when we compare it to mitosis. All right? So we'll be looking at differences between meiosis and mitosis along the way. So let's go. So we'll start with Prophase I. A lot of the events that happen during Prophase I are identical to the events that happen during prophase of mitosis. So we're going to get things like chromosome condensation. Remember, that's just packaging our chromosome into smaller spaces so that we can more easily move those chromosomes around. That happened in mitosis. We're going to have these centrosomes, these yellow structures, move to opposite sides of the cell. That happened in mitosis. We are going to build the mitotic spindle, which is made of microtubules. That's the yellow lines that are growing out of the centrosomes. Remember, the microtubules are what will move our chromosomes around. That's identical to mitosis. But here we get a difference. So crossing over occurs. Crossing over we'll discuss in depth in another video. But crossing over is when these two, what we call homologous chromosomes, combine together, they touch one another, and they actually swap a little information with one another. So that's a complex process. We'll talk about it in another time. But that step happens during Prophase I of meiosis. That does not happen during mitosis. All right, so what else happens during Prophase I? We're going to remove the nuclear envelopes. That happened in mitosis. That will allow us to attach the microtubules to the centromeres of the chromosomes. So microtubules attach to each chromosome at the centromere. And those are the major events for Prophase I. So we'll go straight into Metaphase I after that. In Metaphase I, the microtubules that are attached to the centromere of each chromosome pull the chromosomes to the center of the cell. So that's just like in mitosis. We align the chromosomes along the metaphase plate, but there's a critical difference here. So again, the metaphase plate is where the chromosomes line up. The difference is that the homologous chromosomes, here the blue chromosome and the red chromosome, are attached to one another. And that's a major difference compared to mitosis. So instead of just the sister chromatids lining up along this line, we've got these pairs of homologous chromosomes lining up. All right, so the chromosomes are lined up. We then proceed right into anaphase of meiosis, anaphase I. In this step, these homologous chromosomes that were touching one another, they're pulled apart to opposite sides of the cells. So this is another difference. The homologous chromosomes are pulled apart. In mitosis, we pulled apart sister chromatids. So this step looks different. And we're pulling one homologous chromosome upwards and the other one downwards. The two sister chromatids are still attached, within each homologous chromosome. As soon as anaphase 1 is complete, we can move directly into telophase 1. In telophase 1, it's a lot like mitosis. We're going to make those two new nuclear envelopes to surround the DNA on either side of the cell. But the main difference is we do not decondense the chromosomes. We don't unpack them because we're going to go through these steps again. So cells proceed directly to prophase II. They don't continue into interphase like they did during mitosis. So when telophase 1 is done, we proceed directly into prophase II. So prophase II is going to happen in both of the cells that we've made during meiosis 1. We don't need to condense the chromosomes again because they're still condensed from meiosis 1. But we do need to move the centrosomes apart. We need to build the mitotic spindle, and we need to remove the nuclear envelope so that we can attach those microtubules to the centromeres of the chromosomes. So this is almost identical to mitosis, except we don't need to condense the chromosomes because they're already condensed. When we're done with prophase II, we'll continue straight into metaphase II. In metaphase II, the chromosomes are pulled to the center of the cell because of those microtubules. The chromosomes align on that metaphase plate, so they form this single file line along the metaphase plate. And then here, the sister chromatids are attached to one another, and we've aligned these chromosomes. Metaphase II looks just like metaphase of mitosis in the way the chromosomes are lined up. So this is more similar to metaphase of mitosis. All right, once we've lined up the chromosomes, we can enter anaphase II. Here, we separate the sister chromatids. The sister chromatids are pulled apart to opposite sides of the cell. Here we can see that happening. They make that V shape as they're pulled through the cytoplasm. And that's just like anaphase of mitosis. Once we complete anaphase II, we'll do telophase II. We're going to form 4 nuclei, 2 up here and 2 down here in the 2 different cells where telophase II is taking place. We're going to unpack the chromosomes because we're done with this process. And we will have made our 4 gametes that are genetically different from one another due to the steps of meiosis. So those are all the steps of meiosis. Let's quickly consider the products of meiosis. What have we made? Again, we have made 4 cells. Why did we make 4 cells? It's because we did 2 rounds of cytokinesis. We did cytokinesis following telophase 1 and following telophase 2. So we started with 1 cell, we divided it in half during the first cytokinesis to get 2 cells, and then we divided both of those in half again after the second cytokinesis to make 4 cells. So 2 cytokinesis events explains why we get 4 cells. All right. We're also made haploid gamete cells. They are 1N. Why did this happen? This is because during anaphase 1, we separated apart the homologous chromosomes. So this might be the hardest detail to understand about why the products of meiosis are different than those of mitosis. And we can think about this another time. But the reason these cells are haploid at the end is specifically because homologous chromosomes separated during anaphase 1. All right, what's the other difference that we had for a product of meiosis is that we made these genetically different cells. And there are 2 reasons that our cells at the end of meiosis are different from one another. The first reason, 1, is that crossing over occurred during prophase 1. So again, we'll talk more about the details of crossing over in another video at another time. The second reason that we get genetically different cells, number 2, is that we had independent assortment of chromosomes during metaphase 1. And we'll look at the details of that a little later as well. But those 2 factors contribute to why our products of meiosis are different from one another. So we'll end there. I'll let you ask me any questions you have in class or on the discussion boards, and I will speak to you again soon.